Assalamu alaikum. Dear students, I am here Hafiz Muhammad Ammar, co-teacher microbiology code BUTN 4128. Today is lecture number 15 entitled Chemical Requirements of Microbial Growth. Every organism in its environment must find all of the substances required for energy generation and cellular biosynthesis. The chemicals and elements of this environment that are utilized for bacterial growth are referred to as nutrients or nutritional requirements. There are two main categories of nutrients: macronutrients and micronutrients. We have already discussing in previous lectures. In macronutrients, we will discuss carbon and nitrogen in this lecture. Carbon is the 15th most abundant element in the Earth's crust and the 4th most abundant element in the universe by mass. Carbon's abundance, its unique diversity of organic compounds, and its unusual ability to form polymers at the temperatures commonly encountered on Earth enables this element to serve as common element of all known life. Carbon is the second abundant element in the human body, about 18.5% by mass after oxygen. Most bacteria use some carbon containing compounds as an energy source, and many use carbon containing compounds as building blocks to synthesize their cell components. All organisms require at least small amount of carbon dioxide. However, some can use carbon dioxide as their major or even sole source of carbon. They are also called autotrophs. Some autotrophic organisms reduce carbon dioxide to glucose and other organic molecules. Other organisms require organic compounds as a source of carbon. They are also called heterotrophs. Both autotrophic and heterotrophic organisms can obtain energy from glucose by glycolysis, fermentation, and the Krebs cycle. Carbon and energy sources. All living organisms must require some source of energy. On the basis of this energy source, organisms can be divided into following categories. The organisms that use light as their radiant energy source, they called as phototrophs, and those organisms which use carbon as oxidized form, an organic form of carbon, they called as heterotrophs, or also called chemoheterotrophs, which use this organic carbon, oxidized organic carbon, as a source of energy. These organisms oxidize inorganic compounds and use inorganic compounds oxidation as a source of carbon energy source, that is called lithotrophs. The carbon requirements of organisms must be met by carbon organic form. Organic form of carbon is a chemical compound with a carbon-hydrogen bond, or their source may be of carbon dioxide. Organisms that use organic carbon, they are also called heterotrophs, as we already discussed, and organisms that use carbon dioxide as sole source of carbon for growth, those are called autotrophs. These four categories of the microorganisms on the basis of carbon and as energy sources. On the basis of carbon and energy sources for growth, four major nutritional types of prokaryotes may be defined. As photoautotrophs, their source of energy is light, and their carbon source is carbon dioxide. For example, cyanobacteria and purple and green bacteria. The second one is photoheterotrophs. Their carbon source mainly is organic compounds, and for example, purple and green bacteria. Third one is chemoautotrophs, or also called lithoautotroph or lithotrophs. Their carbon source mainly is carbon dioxide. And fourth one is chemoheterotrophs. Their carbon source is organic compounds. Examples includes most of the bacteria and archaea. Almost all eukaryotes are either photoautotrophic, like plants and algae, or heterotrophic, like animals, protozoans, and fungi. Lithotrophy is unique in prokaryotes, and photoheterotrophy, commonly in the purple and green bacteria, occurs only in a very few eukaryotic algae. Phototrophy has not been found in the archaea, except non-photosynthetic light-driven ATP synthesis in the extreme halophiles. Carbon dioxide. All bacteria require small amount of carbon dioxide for growth. Thus, this requirement is usually met by the carbon dioxide present in the atmosphere, or carbon dioxide produced endogenously by cellular metabolism. Some organisms, such as Brucella abortus, require much higher levels of carbon dioxide, like 5 to 10% for their growth, especially on fresh isolation, like capnophilic. Pneumococci and gonococci are other capnophilic which grow better in the air supplemented with 5 to 10% carbon dioxide. Capnophiles. These are microorganisms that live in the presence of high concentration of carbon dioxide. Some capnophiles may have a metabolic requirement of carbon dioxide, while others merely compete more successfully for resources under these conditions. The term is generally descriptive one and has less relevance as a means of establishing a taxonomic or evolutionary relationship among organisms with this characteristic. In 2004, a capnophilic bacterium was characterized that appears to require carbon dioxide. This organism, Mannheima succini producens, has a unique metabolism involving carbon fixation. While carbon fixation is common to plant life on Earth, since it is the key initial step in the biosynthesis of complex carbon compounds during photosynthesis in the Calvin cycle. It is found in relatively few microorganisms and not found in animals. This can attach carbon dioxide to 3-carbon backbone of phosphoenol pyruvate, an end product in glycolysis, to generate the 4-carbon compound oxaloacetic acid, and immediate in the Krebs cycle. Campylobacter, a type of capnophile, that meaning curved bacteria. It is a gram-negative bacteria. Campylobacter typically appears comma or S-shaped. They are motile. Most Campylobacter species can infect humans and other animals by causing diseases. The bacterium's main natural reservoir is poultry. Humans can contract the disease from eating food contaminated with this species. Another source of infection is contact with infected animals which often carry Campylobacter asymptomatically. A recent study used a novel combination of techniques to reveal how carbon dioxide injections could affect sulfate reducing bacteria that catalyze a key biochemical process in the deep surface. The technique was used in a process that could reduce greenhouse gas emissions to the atmosphere. Ultimately, they found the carbon dioxide sequestration on indigenous microbial populations lead to new strategies for improving success of carbon dioxide sequestration by helping to reduce climate change. Nitrogen, a colorless and odorless diatomic gas with the formula N2. Dinitrogen forms about 78% of Earth's atmosphere, making it the most abundant uncombined element. Nitrogen makes up 14% of dry cell weight, and it is used to form amino acids, DNA and RNAs. So, sources of nitrogen include proteins from most of the bacteria, ammonium that is found in organic matter, nitrogen gas N2 that is obtained directly from atmosphere, important nitrogen fixing bacteria live free in soil or associated with legumes, for example peas, beans, alfalfa and clover. Legume cultivation is used to fertilize soils naturally. Nitrates, salts that dissociate to give nitrates. Nitrogen cycle. All organisms, including microorganisms, need nitrogen to synthesize enzymes and proteins and nucleic acids. Some microorganisms obtain nitrogen from inorganic sources, and a few obtain energy by metabolizing inorganic nitrogen containing substances. In this cycle, you can see the fixation of nitrogen. Atmospheric nitrogen is being absorbed by plants in the form of precipitation or in the form of lightning fixation. If we move towards these lightning fixations, precipitations, the nitrogen absorbed by soil is being fixed by bacteria. There are some bacteria in soil, those are fixing nitrogen to make it available to the plants. If we see organic matter that is decomposed by microorganisms including fungi and bacteria, they mineralize the nitrogen in the form of ammonium. This ammonium is also being provided by industry like fertilizer industries. Those use fossil fuels, and these fossil fuels are also emitting nitrogen gases in the air. This ammonia is being converted again by bacteria's nitrification bacterias, making it nitrites, then convert into nitrates. These nitrates are also being decomposed by the manure coming from dairy and by the action of denitrification bacteria, also coming from being mixed in the water by runoff or eutrophication in the form of leaching. This nitrate form of nitrogen is being uptaken by plants, and the cycle continues. Another example for nitrogen cycle depicts more clear view about nitrogen cycle. You can see plants absorb in the form of nitrates which are converted by nitrifying bacteria conversion of nitrites into nitrates, and these nitrites are being converted by another bacteria from ammonium that is coming from nitrogen fixing bacteria working in the soil environment. These nitrogen fixing bacteria are also attached with the peas roots, legumes roots, and here are the decomposers including bacteria and fungi. They convert plant debris, organic material, into ammonium by aerobic or anaerobic reactions. So here is the clear role of microorganisms in the nitrogen cycle, and this cycle also shows how important nitrogen and the role of microorganisms being played in the atmosphere. Fastidious bacteria. Some can synthesize all 20 amino acids found in proteins, whereas others must have one or few amino acids provided in their medium. Certain fastidious organisms require all 20 amino acids and other building blocks in their medium. For example, Spiroplasma species, those are tiny spiral bacteria that lay cell walls, are among the most nutritionally fastidious organisms known. Recently, Kevin Hackert, a US Department of Agriculture scientist, devised an extract formula of 80 ingredients, including lipids, carbohydrates, amino acids, salts, vitamins, organic acids, and penicillin, to meet their needs. In this laboratory, he uses his medium to keep more than 30 species of spiroplasmas alive, and while making it possible for researchers to study them outside more than 100 insects, ticks, and plant species that may normally inhabit. Until now, most spiroplasma species have been impossible to keep alive outside their hosts. United States Department of Agriculture scientists are now trying to formulate complex media to grow mycoplasma-like organisms, a group of bacteria that also lay cell walls. These bacteria causes hundreds of crop diseases and millions of dollars in economic losses each year. They are spread from plant to plant by infected insects. Another medium being designed would grow the bacterium Mycoplasma pneumoniae, which is the cause of a form of walking pneumonia in humans. Medical researchers are particularly interested in one species that is experimentally causes tumors in animals. Another species kills honeybees, and a third lives harmlessly in the Colorado potato beetle, an insect that damages potato, eggplant, and tomato plants. Scientists hope to alter this last species genetically so that it can kill its potato beetle host. Many disease-causing organisms obtain amino acids for making proteins and other nitrogenous molecules from the cells of humans and other organisms they invade. Once amino acids are synthesized or obtained from the medium, they can be used in protein synthesis. Similarly, purines and pyrimidines can also be used to make DNA and RNA. The processes by which proteins and nucleic acids are synthesized are directly related to the genetic information contained in a cell. So here are the reference books where from the material was used for the preparation of this lecture. You can also read these books form as available as a PDF form in the Google class. Thank you. Hope you all understand the role of carbon and nitrogen in microorganisms growth. Thank you very much.